Amy here, back with the last video for Chapter 7 over photosynthesis, on which we will be looking at other types of photosynthesis that some plants show. The majority of the plants that we talked about, and I mentioned this in the other videos, are what we refer to as C3 photosynthesis. In other words, in the Calvin cycle, when you had RUBP, and we joined in CO2, that made a 6-carbon substance, that immediately broke down into 3PG, which was 3 carbons. So that's the first this is the first recognizable thing that we see in the Calvin cycle. So we call it C3 photosynthesis. However, in hot, dry climates, if you think back to the structure of the leaf, the CO2, this right here, is taken out of the air and in through the leaf through the openings called the stomata. Well, if it's really hot, the water will evaporate out of those stomata, causing the plant to wilt, and possibly die, dry up. So in order to avoid wilting and losing too much water, plants close the stomata. However, when that happens, photosynthesis is usually hot. It's very sunny as well. The amounts of CO2 start to go down. And remember, oxygen is a byproduct from the water splitting. The amount of O2 increases. When oxygen levels get too high, oxygen will start combining up here with RUBP. RUBP will start combining with oxygen, leading to the production of CO2, but you're reducing the amount of RUBP. And this is called photorespiration, and it really bogs down and slows down the Calvin cycle and how that can happen. But plants need to do that so they don't dry up. There are some plants that have evolved this way of doing C4 photosynthesis. Now, what they're going to do is take CO2 and fix it to PEP, which is a C3 molecule. So here's a carbon, here's 3 carbons. You combine them together, which is going to make oxaloacetate, which is a C4 molecule, and then be stored as malate or malate. And this helps the C4 plants avoid photorespiration. And they're actually going to do this by space. So we're going to look at the difference in structure. How do C4 plants, how do they set this up so that the CO2 or the increase of oxygen cannot get to the RUBP? We look at the next slide. We'll see structurally how they do that. On this plant, here's our C3 plant. Here's the cuticle, the epidermis. Right here, or this would be the cuticle. This would be the epidermal cells right here. This would be the cuticle, the waxy cuticle. And then down here is our stomata, or our stoma. This is where oxygen is going to come in here. And then inside here, we said the photosynthesis takes place in the mesophyll cells here. And then your leaf vein where water is coming up. Here's the xylem. Here's the phloem right here. They're taking the sugars back down. You have these bundle sheath cells around that leaf vein. But photosynthesis is taking place all through here in the mesophyll cells. So if these things close and the O2 levels build up in here, O2, 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 you can see where that oxygen will combine with the RUBP and cause photorespiration. What a C4 plant does is it organizes the leaf a little bit differently. It puts the mesophyll cells around, right here, around the bundle sheath cells in here. Now notice these bundle sheath cells, have chloroplasts. So what's going to happen is when this has to close, oxygen will build up out here. Okay, oxygen will build up out here. But the Calvin cycle, okay, will take place in the bundle sheath cells in here. So Calvin cycle is happening in the bundle sheath cells. Okay, this little bit of CO2 that's left out here will be attached to by PEP, okay, to make oxaloacetate, okay, which is then taken into the bundle sheath cells. Okay, so the Calvin cycle is happening in here, and it's sort of protected from all the C4 plant. Kind of helps partition CO2 fixation, okay, by space. Okay, by space. The way that it organizes the leaf, it keeps the oxygen away from the RUBP and avoids photorespiration from happening. Okay, this is just more of a simplified version. Okay, here is your C3 plant. CO2 comes in the stomata, and it goes through the Calvin cycle right here, 3PG, G3P, RUBP. But as the oxygen builds up, as the O2 builds up around the mesophyll cell, that O2 can bind to RUBP and shut down this pathway. Okay, C3 plants would be a lot of the things that were most plants, things like maple trees, tulips. Sometimes when you're talking about crops, this would be your wheat, rice, oats, those kind of crops. And in our lawns, if you think about a lawn, earlier in the summer when it's cooler out, a lot of sunshine, but it's cooler. There's a lot of water, rainfall from the spring. Your grass, you have these real fine bladed grass called Kentucky bluegrass that we use on a lot of our lawns and yards. Okay, and it's really lush and it's really green. Okay, as the summer progresses, okay, and it gets hot and dry. So you get into July, August, it's really hot. There's not as much moisture in the ground. What happens to the Kentucky bluegrass? It gets brown, right? Your yard gets brown and crunchy. Okay, you look at what happens to wheat. Wheat tends to mature the 1st of July, and then it really dries down. Same thing with the oats. Okay, over here this would be the C4 plants over here. Okay, this is when they really start cranking it up. Okay, because when it's hot and dry, notice here is the mesophyll cell. Okay, and the CO2 is going to be taken in by PEP and made into oxaloacetate, okay, which is then actually reduced to malate right here. And then they take the malate into the bundle sheath cells and drop off the CO2. And then the C3 molecule will come back and can get some more CO2. Okay? Or not the CO2 molecule, the C3 molecule can come back and get some more CO2. Okay, plants in the C4 category include things like corn. Yeah, you often hear farmers say, oh yeah, this heat is good for the corn growing. Okay, well, actually they are about 2 to 3 times more efficient than the C3 plants when it's really hot and dry like that in the summer. And they will still keep growing like crazy. In our yards, you notice when the Kentucky bluegrass starts to get all brown and crispy, what takes over? Crabgrass, right? That thick-bladed, doesn't feel as good on the bare feet. You get this real thick-bladed crabgrass that's still green in July and August. So your yard is brown except for a few of these weeds and crabgrass that are growing up through. And they're out-competing the Kentucky bluegrass because being a C4 plant, they are better adapted to that hot, dry environment later in the summer. The second type of other type of photosynthesis is what we call a CAM photosynthesis. Okay, CAM is Crassulacean-acid metabolism. Okay, and what happens is this is carbon fixation by time, whereas C4 plants were by space. You had the bundle sheath cells, The mesophyll cells that protected a barrier had a space between the oxygen and where the CO2 was being fixed to RUBP. CAM plants will actually fix the CO2 at night. Okay, they will fix the CO2 at night. So during the day, when the light reactions are cranking out the NADPH and the ATP, the stomata can stay close so that they don't lose water. And then the C4 molecules, again, which tend to be reduced into malate, can be released to the Calvin cycle then during the day. So the stomata can stay closed when it's really, really super hot, and they will not wilt. And then they will have all this carbon stored up as malate that they fixed during the night. Okay, so CAM photosynthesis will partition that carbon fixation by time of day. Again, this is just kind of the overview of a CAM plant. Where here at night, the stomata will open, CO2 is taken in, okay, is stored. And then during the day, when it's hot and dry, then that carbon is released as carbon dioxide, and then the C3 molecule is stored again for overnight for capturing more CO2. Okay, these tend to be in very hot, arid climates. The example they're showing there is pineapple. A lot of you are into raising succulents. A lot of the plants that we consider succulents would go through CAM photosynthesis. And actually some very common plants as well, like tulips, for example, can do CAM, Crassulacean-acid metabolism as well. So just kind of the overview, the advantages and disadvantages of these three types of photosynthesis. C4 plants, they are better adapted to having like high light, high temperature, and being a fairly dry environment. These things will outperform these 2 to 3 times the rate of a C3 plant. Okay, whereas C3 plants are better adapted to cooler, wetter, more think springtime in Ohio type environments. And then finally, CAM plants occur in some flowering plants and then some non-flowering plants like succulents and pineapples, those kind of things. An extreme aridity, okay, deserty like chaparral, dry shrubland, very dry environments where it's very, very hot and very, very arid or dry. The last slide that I kind of want to go over, this is not in your textbook, but I do want to talk about in case you see some of these graphs, and I'm not sure why the textbook doesn't put it in there, but I do want to make sure that we cover these, and we also do a lab in class where we look at some of these factors as well. There's 3 of them. The first one, what would affect the rate of photosynthesis? Well, what does photosynthesis mean? You look at the root word photo, it means light. So obviously, the greater the light intensity, the higher the rate of photosynthesis. Now you'll notice that curve is up to a certain point. Once you reach a certain light intensity, you'll kind of max out, where those light-dependent reactions are taking place as fast as they can. The Calvin cycle is using up the NADPH and the ATP and recycling those back as fast as they can. But so they sort of max out. The second thing, what else does photosynthesis need besides light? Also water. Obviously, if there's not enough water, that will affect the plant, but also CO2, carbon dioxide concentration. Okay, similar idea. Increase the amount of CO2, it will increase the rate of photosynthesis to a certain point when those enzymes are working as fast as they can, and they're sort of maxed out. The accelerators stomp to the floor, and they're at top speed here. Okay. Third one has to do with enzymes, and that's temperature. All right. So you increase the rate of photosynthesis by increasing the temperature, more molecular collisions, you're speeding up the molecular motion. There's more collisions between enzymes and substrates for all those different steps in the light-dependent and light-independent reactions. Notice that it doesn't max out, though. you hit this peak usually between 35 and 40 degrees Celsius. We talk about that in the enzyme chapter. You start to get too warm. This is where your denaturing of the enzyme comes in, and it's going to be less effective, and the rate of photosynthesis will die off. Okay, so in case you see these graphs again, I wanted to make sure that you are familiar with these three things and why they look a little bit different. Other than that, that pretty much wraps up photosynthesis for us here in AP Bio. If you have any questions, again, please make sure to write them down, ask me in class. Otherwise, this is Hammy, out.